All right, so the last unit was largely focused on central dogma, how we go from DNA to get RNA, and how we go from, actually, I should say DNA, we get more DNA through replication. How from DNA we can get RNA through transcription, and from RNA to protein, we get protein through translation. What's important to note is that there are things that elaborate further on the central dogma that can be a bit complex, but are actually really important fields, really important concepts for us to know, as well as current active areas of research, particularly one of the concepts, and you'll appreciate why given how complex it is. But we'll be talking about all of this against the backdrop of a couple of syndromes called Klinefelter syndrome and Down syndrome. First, Down syndrome. I'm sure most of us have had people in our lives who have Down syndrome. It's a genetic disorder that has a wide range of developmental delays as well as physical presentations. Some of the physical presentations include brachycephaly, which is usually a bit of a shorter head. Usually, there's also brachydactyly, which is also shorter digits. Bless you. Shorter digits or shorter fingertips usually. Duodenal atresia, which is usually this folds the eyelids that usually cause a bit of a slant in their eyelids. Yeah, they have an upward slant, and that's usually because of the epicanthal folds within their eyelids. There's also sorry, I'm all over the place today. I'm severely undercaffeinated. I'm just take a step back. The epicanthal folds are what the folds in the eyes that cause this upward slant. Duodenal atresia is developmental closure of the duodenum within their GI tract. There's also a fifth finger clinodactyly, which is usually like a shorter fifth digit. There's also hypotonia, which is lower muscular tone, particularly when they're first born. And that is just lower muscle mass, which also leads to more lax ligaments. There's also the intellectual disability, but in addition to that, there's also the last two physical presentations are that there's usually a shorter stature and there's a larger gap between their first two digits on their feet. We're going to talk a bit about the etiology of Down syndrome in a moment. Klinefelter syndrome. It's the most common genetic cause of male infertility. What's really unique is that because of how many different causes there are for male infertility, up to 65% of the cases of Klinefelter syndrome can go undiagnosed. Some of the key symptoms that are associated with it are gynecomastia, which is usually the developmental of male breast tissue. There's hypogonadism, which is an underdevelopment of the sex organs. And one potential diagnostic way in which this has been identified is via elevated levels of follicle stimulating hormone. It's released by the pituitary gland, and it's important for both spermatogenesis and oogenesis, the development of sperm and eggs. And it's also used oftentimes for identifying general infertility, the elevated levels of FSH, follicle stimulating hormone. What's important to note here is that the epidemiology and incidence are both fairly similar. The current prevalence rate for both is about 250,000 in the U.S. alone. Klinefelter being a male infertility disease, it's 250,000 men in the U.S. And the incidence rates are, again, pretty similar. Both appear to occur randomly. There's no known actual behavioral activity or environmental factors that seem to cause it. One association with it, at least that's well known or well appreciated with Down syndrome, is age. The likelihood of Down syndrome increases as the age of the mother increases. And the Similarly, there's an association with age, but with Klinefelter, but except with the father. The association, the likelihood of Klinefelter syndrome goes up as the age of the father goes up. So for etiology, both of these are caused by defective meiosis and aneuploidy. So meiosis is, so we're, I hope we'll get the chance to talk about it. I don't know if we will, but so mitosis is basically cell cycle progression, cell proliferation. Meiosis is that for specifically for eggs, for an egg and a sperm. And so what ends up happening here is that we have all of our cells are diploids. We have two copies of our chromosomes. Our cells that undergo meiosis, they usually have one copy. During meiosis, they'll split and have one copy of each. They'll have, instead of 2N or two copies of chromosome, they'll have 1N. And what ends up happening is that there's some sort of defect that causes an extra copy to occur. And that's what aneuploidy is. That's aneuploidy is typically defined as having extra copies of chromosome, more copies of chromosomes than you're supposed to have. And so the way in which that manifests here is that with Down syndrome, there's an extra chromosome 21. And so another name for Down syndrome is trisomy 21. That means there are 3 copies of chromosome 21. Klinefelter syndrome is usually one or more extra of the X chromosome, of that sex chromosome. So this chromosomal aneuploidy, again, which is having aneuploidy having extra copies of these chromosomes than you're supposed to have, it usually results in what we call an elevated gene dosage. So if there are more copies of these chromosomes, that means there are more copies of these genes. That means there's more gene being transcribed, and subsequently more protein. So if we have 3 copies instead of 2, you can imagine that we'll approximately have 50% more transcription, 50% more protein being translated from having 3 chromosomes instead of just 2. But during the course of the Down syndrome versus Klinefelter syndrome description, the details of their symptoms were quite different. And so if both of them are an example of aneuploidy on different chromosomes, one question to keep in your mind that we're going to talk about at the very end is, why is the severity of the two so different? And so that's a question I want you to hold on to that we'll talk about at the end. And it cannot be explained. One thought might be that it might be explained by the different size of the chromosomes. And it cannot because chromosome 21, the chromosome that undergoes aneuploidy during Down syndrome, Down syndrome does not have a more severe presentation because of chromosome 21 being bigger. It's actually smaller, for about 48 megabase pairs and approximately 250 genes, whereas the X chromosome is much larger. And so the size does not explain the difference, the clinical difference between these two syndromes. So we're going to define something that's a bit of a controversial thing to define. If you talk to, in your spare time, if you talk to a geneticist, if you talk to a geneticist who's probably older than 50 years old, they're going to have one definition. And if you talk to someone who's under 50 years old, they're going to have a different definition. And the two of them will probably bicker and fight for an unnecessarily long amount of time about what exactly epigenetics is. But typically, epigenetics occurs at the level between DNA and RNA. And a traditional older definition of epigenetics is that it's a series of stable or heritable cellular modifications that can impact the genome regulation, but does not alter the genome sequence itself. So it's not, what's happening here with epigenetics is that we're not altering the actual base pairs, the nucleotides here, but rather these like stable and inheritable changes that impact its regulation. The more modern or newer take on how we define epigenetics is rather that there are these like structural changes that happen on chromosome regions that can actually alter and perpetuate altered activity states, meaning it can either go alter the regulation to be on or off, and The reality, I would say, I'm not a geneticist, nor will I ever be. But if I was to have a take on this, I would say it's probably more likely a mix of the two would probably be a more appropriate way to try and think of epigenetics. One thing that always helps me whenever I think about epigenetics, even though it's like this like really broad and amorphous term, epi means like next to, and genetics, and genetics is usually genes. And so usually epigenetics are these things, these modifications that happen in next to or in or around our DNA and our genes. And so you'll appreciate what that actually looks like when we get further into this lecture. Yeah, I was going to stop for questions. Go ahead.
Do you know like what the main argument that they have is?
Great question. So some epigenetic changes are, I actually should have taken a moment to actually try to highlight the differences. The traditional definition is that these epigenetic changes that occur like in and around our DNA are stable. They will not like revert back, and they are inheritable, meaning they'll be passed down to your progeny. A more modern definition where we have the tools to see the resolution of like these epigenetic changes happening in real time. Some of these changes might happen and then might revert back or go away. And so we're going to talk about some examples of the two, and which is why I kind of think it's actually a mix of the two. But there are changes that can happen that are stable, that don't change, that are actually passed down to future generations, and others that are more transient and more dynamic. Does that help clarify that? Thank you for that question. I appreciate that. So examples of a stable and heritable epigenetic change. There are, as you've probably been able to pick up by now, there's so many different kinds of mice that we use in research, and they all have different names. There is a mouse called the agouti mouse, where their yellow coat color is regulated by a specific gene locus called the agouti gene locus. And what's really fascinating is that in the agouti mouse, that coat color can change simply by altering the diet of the mother during pregnancy. And so the gene that regulates this yellow coat color is altered and modified in response to some factor, in this case, the mother's diet, and it creates a stable change that's then passed down to future generations. And so this is one example in which epigenetic changes are stable. They won't change, like the coat color typically won't change for these mice unless, again, there's, for a single mouse, their coat color won't change. It'll change because of the diet that the mother had during pregnancy. There's also a pretty similar and more upsetting example where during World War II, there were tactics used to starve out villages. And there were examples of some of these different communities where the mothers were pregnant during this period of starvation. The future generations had epigenetic changes that occurred that predisposed them to higher degrees of obesity and a general shorter lifespan. Another simpler example is smoking tobacco. There are changes that happen in and around our DNA that can then be passed down to future generations that actually predispose them to developing allergies and asthma. An example of the more transient, dynamic, or plastic epigenetic changes, a really simple one is our circadian rhythm. There are a series of genes that appear to be regulated or controlled by our circadian rhythm. And you can actually see a dynamic of like 12-hour changes in gene expression. This is a totally very active area of research that we're still understanding what genes or what things are regulated by circadian rhythm. And we've identified specific transcription factors and proteins that seem to be circadian clock transcription factors. But it appears that there are some modifications that happen to our histones and nucleosomes, and these are structural titles that were discussed in the pre-lecture that where the nucleosomes and histones can be modified, and you can see the changes of that over the circadian rhythm. That's what's depicted here. Furthermore, in the context of neuroscience, there's a great degree of plasticity with our neurons, and you can actually see specific transcription factors. An example here is CFOS, where there are regions of our chromosomes that can become open, and the CFOS can engage during learning, but that can quickly go back and be reverted in examples of forgetting, which I'm sure we're all familiar with and sometimes frustrated by when we have an exam and we forget something. But the key thing to note here between these two slides, going back to the question we had earlier, is that epigenetic changes can be stable, permanent, and heritable, or more transient and dynamic. And the mechanisms by which that's controlled, or we're going to talk a little bit more about that, but it's still an active area of research. I'm going to stop there and give space for a couple questions.
So with the neuroplasticity, I guess I don't understand how regulating chromosomes, like are neurons being created with neuroplasticity?
Great question. So I'm gonna preface this with a caveat. I think neuroscience is incredibly complicated, so I'm going to give you like a very reductive answer based on what I understand. Whenever people in neuroscience talk about things like this, they usually talk about the circuitry of a neuron. That's usually what they talk about, I think, when they're describing like certain signaling pathways and the way genes are controlled. And so what they're talking about here is that they've identified certain regions of the genome of our DNA that are associated with learning. And so here, this transcription factor CFOS is associated with learning, and when it can bind to its specific target region, that's associated with learning. That basically learning is occurring, and memory is formed during this process. But there are times where we've learned something, but we forget it, and so that's associated with CFOS no longer binding to and interacting with that specific site of the DNA. And so this ability for this transcription factor to go in and bind and to leave, and how easy it's able to access that region of the genome. So these two nucleosomes are separated, enabling CFOS to come in. But if they're closed, CFOS can't go in, and that's, for lack of a better phrase, forgetting, where that transcription factor can't contribute to the circuit that is contributing to learning. Does that help clarify? Make sense a little more? Okay. Yes.
For the two definitions and the thoughts on how genetics work, was there more evidence in the past of the stable heritable and permanent? And then we learned more information. Why haven't we adapted to understanding that it's more transient than it's not heritable? We have more information, or are both true at the same time?
Great question. So we only had access to certain tools that enabled us to see permanent changes, but as science, as scientists developed better tools that can see with finer resolution things going on and off, we're able to see those changes more transiently. But there's a non-scientific answer to your question where scientists care a lot about what they've proven, and they don't want to let go of what they've proven. I'm not trying to disparage. I feel like I'm disparaging older scientists here. I shouldn't be doing. That's not what I'm trying to say. But there's also a human element to scientists where they've devoted their entire career to this idea, and they're a little reluctant to give that up. And so, typically, older geneticists, when they talk about epigenetic changes, it's that traditional definition, which isn't to say that their findings were wrong, but in the context of some other things, maybe their contributions can help contribute to a definition of epigenetics, but doesn't define everything. I hope that was a long-winded answer, but I hope that helps make sense of it. Yes.
So in the last slide, it was examples of stable epigenetics. It's considered stable because once the offspring is born, they're like co-colors.
Right.
But
Yeah.
if it's not stable because in one organism's lifetime, like their co-color can change. It's like.
If coat color was a transient, yes, exactly. Yeah. All right, so we're going to go ahead and press on. Okay, so there are going to be two primary mechanisms of epigenetic changes that we're going to be talking about that control chromatin accessibility. This was talked a little bit about in the pre-lecture, but the neuronal learning slide back here is helpful, where chromatin accessibility is like the opening. The chromatin is the packing of our DNA around these histones to form nucleosomes. Chromatin accessibility is like the opening and closing, the moving of these nucleosomes to make regions of our DNA accessible for transcription factors to come in and bind. So there's going to be two ways that we talk about chromatin accessibility being changed. The first is going to be DNA methylation. That's going to be the addition of a methyl group directly onto our DNA. It's not changing the nucleotides or the sequences here, but it's coming and altering next to the DNA. And typically, when DNA methylation occurs, that's going to induce the compaction of our chromatin. So it being compacted makes it less accessible for a transcription factor to come in. And so because of that, that's going to repress gene transcription locally where that DNA methylation is occurring. And so again, a key thing to note here is that DNA methylation, it's in the name, but it's really important to note here. DNA methylation occurring is occurring at the DNA level. The second mechanism we're going to talk about are histone modifications. It was in the pre-lecture, but again, histones are these proteins that our DNA is actually wrapped around that make up the nucleosome. So there's four different kinds of histones: H2A, H2B, H3, and H4. And you'll have two of each. So you'll have this octamer of histones with DNA wrapped around it. That makes up the nucleosome. And so what ends up happening is that on our histones, there can be modifications that can the histone is a protein, so this would be a post-translational modification to it. And so what's important to note is that these modifications come with a wild degree of complexity that we will quickly see the magnitude of that complexity. But some changes will induce chromatin compaction, others will open it up. And if that opens up the chromatin, that's making regions of the DNA more accessible, which enables transcription to occur. And so that means that histone modifications can either activate or repress gene transcription. For DNA methylation, again, that typically induces gene silencing. What ends up happening is that it's typically on a cytosine, and there are specific enzymes that will induce this. The enzyme is called DNA methyltransferase. So it's a DNA that is transferring over a methyl group onto our nucleotide. In this case, it's cytosine. And so that's going to induce this 5-methylcytosine. Now, there are mechanisms in place that can help that enable for this methylation to be reversed. So like this is an example where historically DNA methylation was seen to be a permanent modification. There are mechanisms that we've learned where this can be reversed. And again, this is like another example where a biology class is a class of half-truths, where we thought one way, we think this is true, and then 10 years later, we discover this is actually not true, and it's less permanent. And so there's an enzyme called TET. You can just imagine that as just a demethylase, whereby it's working to try and remove that methyl group. So what ends up happening is that the demethylase, that's its goal, but what it's doing is it's adding, it's a dioxygenase, so it's adding oxygen groups here where the methyl group was added. And so there's going to be further modifications that eventually, using our base excision repair machinery, will get it back to a cytosine. And what I should say is that the substrate that's the methyl donor when we go from cytosine to 5-methylcytosine is called SAM, S-adenosyl methionine. That's the methyl donor that DNA methyltransferase uses to add on the methyl group to cytosine. there's a lot of pieces here, a lot of things to try and focus on. What I want to draw your attention to, as is highlighted in blue here, is that the key enzyme that's mediating this methylation is DNA methyltransferase. S-adenosylmethionine is the methyl donor here. And so those two pieces mediate the addition of the methyl group, and TET is what's working to help remove that methyl group eventually by helping to cut out, using our base excision repair and the polymerases involved, to cut out that methylated cytosine and replace it with the appropriate cytosine here. We talked about our CPG islands, these regions of our DNA that are rich in cytosine and guanine. And you can see here that in the CPG islands are typically unmethylated. That'll enable, or less methylated, I should say. There's more gene transcription that'll be occurring there, whereas the heavily methylated regions, heavily methylated CPG islands, will have gene expression repressed there. So one way in which this is really key in biology is when these DNA methylation events that occur over the span of our lives can actually be erased and reset before meiosis. This is called a process. The process here is called genomic imprinting. It's a little complicated, but I'm gonna try and walk us through it and make sure that I'm highlighting the key take-home points so that we're not distracted by all the details here. So to try and help break down some of the colors here. So we have two primordial germ cells here. We have the egg and the sperm. Right now they're diploid. They have two copies of every chromosome. This is pulled straight from a journal that was really helpful in trying to identify some of the key mechanisms here. So that the circle color designates like the environment that it's in, meaning that this is an egg in pink and the sperm is in blue. The regions that have that are under control, the regions that are inherited from the mother and the father are labeled in pink and blue on the chromosomes themselves. There we go. And the regions that are white are unmethylated, and these black lines are going to be methylated regions. So there are going to be methylation events that have happened where there's going to be a total erasing of them leading into meiosis. And then simply by nature of whether it's an egg or a sperm, there's going to be specific methylation events that are innate to it being an egg or a sperm that then during fertilization are imprinted and passed down to the zygote that gets a copy from each gamete or each haploid cell here. And so during development, the original methylation events that are inherited here are inherited simply by nature of coming from an egg or a sperm. And so that is the imprinting part. So the erasing is all the methylation events that exist are first erased leading into meiosis. And once they split into their haploid cells where they have one copy of each chromosome, there are methylation events that occur on each chromosome that are simply by nature of whether it's from an egg or a sperm. And so what ends up happening is that there are going to be specific methylation events that are only passed down from each parent. And so what this looks like, an example of this is, what I should also say is that this is not for every single gene. We have 25,000 genes in our body. Right now, this genomic imprinting has only been associated. Right now, the list is about 300 genes whereby this is controlled. So it's not all genes. But one example is the IGF-2 and IGF-2 receptor gene, where the IGF-2 gene is paternally expressed. The methylation events that occur during genomic imprinting make it so that IGF-2 is only coming from one parent. Similarly, there are methylation events that occur that make it so that IGF-2 receptor, IGF-2R, is only coming from the other parent. And so there's a theory, it's a bit of an antiquated theory, that makes sense in the context of the broader animal kingdom and mammals. It's kind of odd to apply it to humans, but basically, when you think of it across all mammals, particularly placenta-bearing animals, the idea is that IGF-2. We talked about this during the steroid lecture, where that is what the androgen receptor is. That is the gene that the androgen receptor is targeting to promote its transcription. And so IGF-2 is associated with protein synthesis, with protein anabolism, and so it's associated with growth. And so IGF-2, as it's paternally expressed, is meant to promote offspring growth, that it's a bigger, stronger progeny. Whereas IGF-2 receptor is a decoy receptor for IGF-2, meaning that it's basically a sink for IGF-2, taking it away from actually carrying out its biological function. And the idea is that it's meant to restrain offspring growth and ensure that typically during pregnancy, offspring is taking away nutrients or nutrition from the mother. And so the idea here is that IGF-2R is meant to try and restrain offspring growth. The key here is not for you to take home this kinship theory idea, but rather to try and note that this genomic imprinting is a way in which a few hundred genes receive their methylation events, and that there is actual specific biological relationships between the genes, between some of them, that explain why this is happening, perhaps. I'll actually stop there and wait for a couple of questions, because I think this might be a hard slide. I'm going to go here and there. Go ahead.
So, what is being the race exactly and it's still being passed on?
Sorry?
What is being the race that where the network events are still being passed on?
Great question. So, the DNA methylation events that so during spermatogenesis or oogenesis, before we have an actual mature sperm or egg, we have our primordial germ cells. So they are diploid. They have 2 copies of every chromosome. Before meiosis begins, where we get a mature sperm and egg, which are only going to be haploid, which have only 1 copy of each chromosome, all the DNA methylation events are going to be erased, particularly the ones that are inherited from the parents. And what's going to end up happening is that we're going to reestablish specific methylation events that are going to be associated with either it coming with that haploid cell either being an egg or a sperm. So there's going to be an erasing of historic methylation events and new ones established simply by nature of whether it's a sperm or an egg.
Are these new reestablished methylation events specific? Like, do the same ones happen every time? Based on what's a or egg?
Yes, as far as we know. This is still an active area of research, but of the few hundred genes that we know, that's how their methylation occurs.
The methylation events are not. We see the same set of methylation events.
In the context of genomic imprinting, these are inherited because you're inheriting a copy from each parent.
But historic methylation events.
Historic are not passed down.
Once that game will get
For these few hundred genes. There are other genes where there are methylation events that can perhaps be passed down. So we're talking about specifically the genes that apply to this concept.
Thank you.
Yes, Tommy?
The IGF-2 that's internally expressed, there's a methylation event in IGF-2 for the mom, so that it's not expressed.
Exactly right. Yeah, I should have. I'm glad you asked that. I should have said that more clearly. The methylation events that occur in IGF-2 are on the maternal side, such that you're only going to be receiving that copy of that gene from the father.
Don't you still have it, but it's just methylated?
Yeah, exactly. Yeah. Well, so when I say, if I said don't, let me get close to my computer. If I say don't have it, what I should have meant is it's methylated, and so its expression is suppressed. Yes.
So like the sperm and the egg, copies is the same as the parent. So the pattern of methylated, unmethylated, same as the one. So is that on your parent, or is that like you that like one?
Yeah, so your question is different, and you're actually getting at something that I don't know the answer. I suspect that I'm gonna look into this, but I suspect that whatever perhaps degree of methylation for those I'll say as for those genes that this concept applies to, there's probably going to be varying degrees of methylation across everyone in this room on these genes that we're talking about here. And so I suspect there will still be a let's see what it says. I suspect there will still be a difference across individuals, but the general rules that that control that are the same for everyone. But there could still be like differences in like degree of methylation, which I think is what you were getting at. Did I hear that correctly? If I didn't hear that correctly, you can absolutely say that.
Yeah.
Well, I was going to say, I actually thought it was a good question because I actually think you're touching on something that's important that I don't know the answer to. I can venture a guess based on what I know about the concept, but I'm going to look into that more because I actually think that's an important question that I don't know the answer to about this. Yes, go ahead. We'll do one more question.
So the sperm and eggs have different sets of resetted methylation steps.
Yes.
Then the offspring, will they then express a mixture of those methylation events when their own gametes are resetted?
The offspring will have those methylation events as their so this goes, I think, to what Maximus' question was getting at. They're going to have those methylation events from each parent, but perhaps the degree of methylation, because maybe a good way to say this is that the genes whose methylation follows genomic imprinting, follows these rules, there might be perhaps different degrees of methylation between sets of parents. And so there might be different degrees of expression of IGF-2 or IGF-2 receptor across individuals, but the general rules that regulate their DNA methylation applies here. Does that make sense?
Well, I'm trying to ask it. So if I have a certain set of modifications on the eggs, the offspring, the its own eggs would they display the same modifications? Or they display the extra modifications?
Same modifications.
Okay.
Yeah.
So same.
Yeah. All right. So that was DNA methylation. So for histone modifications, they are what dictate actual chromatin accessibility. And so I'm going to preface this by saying, I'm not going to ask you all to memorize this because this is wildly complicated. I still mix things up with this, and you'll appreciate why I'm saying I'm not going to ask you all to memorize these changes, because the numbers are quite astronomical in terms of like what are the possibilities here. But the nomenclature, like the shorthand we use for this that are highlighted in blue, those are going to be things that I'm going to ask you to remember. And so typically these histone modifications, these are post-translational modifications. An example here is we have lysines on this histine that are acetylated. And so we have an acyl group that's added here. And so we have histone acetyltransferase that adds that acetyl group. And we also have HDACs, these histone deacetylases that remove the acetyl group from these amino acid residues. On this slide, we're looking at lysine specifically, but I'll describe a little bit more about some of the other possibilities here. So what's important to note here is that when you add an acetyl group here, it's going to be changing the charge on the histone. It's going to be eliminating the attraction. It's going to neutralize the charge, and it's going to change how these tails on the histones here behave, and it's going to end up opening the chromatin more. So one example of a histone modification in this example here is that a lysine being modified where an acetyl group is being added. That's going to open up the chromatin. The deacetylase is going to close the chromatin as it removes that acyl group. The shorthand here, the shorthand we use here is first is what histone is being modified, so it's H3 here. What amino acid in its position, so it's lysine 9 on this amino acid sequence here, and it's being acetylated. So this is H3 on the H3 histone, lysine 9 has been acetylated. That's the shorthand. And so you'll see a couple other examples to try and appreciate other versions of that shorthand. There's lots of different modifications that can happen, and that they all happen on different time scales. So acetylation is one that happens that's associated with active transcription. It happens on pretty quickly, on the order of minutes here. Another example is phosphorylation. We've already talked about some of the different amino acid residues that can be phosphorylated. We have serine, threonine, and tyrosine. And so phosphorylation is also pretty active. It happens pretty quickly on the order of minutes to an hour, on the order of minutes to hours. An example of what that might look like is H3, histone 3, on serine 10 is phosphorylated. The last thing is we talk about DNA methylation. We can also have histone methylation, and that happens on the order of days. It's a much slower response, but the effect it has is pretty confusing, to put it lightly. What I should say is that the phosphorylation event also adds a negative charge, which is the nature by which it alters the chromatin accessibility. But specifically with methylation, we have histone methyltransferase that adds the methyl group here, and we have HDM, histone demethylase, that removes it. The examples of how the shorthand we use for methylation is, for example, we have on histone H3, on lysine 4, we have a few methyl groups that have been added. That makes it more active. On the same histone at lysine 27, 3 methyl groups are added, which makes it inactive. And so there's a lot of combinatorial possibilities that makes it really hard to track what these different modifications do. And so I'm not asking you to memorize what different methylation events yield on histones. I'll ask you to note what DNA methylation does. There's that one's a bit more clear, but rather what I want you to note is for the kinetics of these modifications, like what timescales they happen at, the shorthand as well, and as well as these enzymes that are involved with the addition of these methyl groups, these acetyl groups. So reading out the histone code are all the modifications that happen across all the histones here. And that possibility is incredibly large because of how many chromosomes we have, because of how many nucleosomes are on each chromosome, and because there are 8 nucleosomes on each, or 8 histones on each nucleosome. The combinatorial possibilities of this are quite large. And so it becomes really challenging and hard to study, but it's very much an active area of research. As new tools emerge, it's becoming a pretty exciting field of study. But to help you appreciate this is just one snapshot of on one nucleosome, all the different possibilities of the modifications that can occur, whether it's on specific lysine residues that are being methylated, acetylated, or serines that are being phosphorylated. For each of these, this is just on one nucleosome, the possibilities that can occur. And if you multiply this out beyond, out to how many nucleosomes are in a single chromosome, this becomes an astronomically high, one of the highest numbers out there. So naturally, I'll ask you to memorize all of that. That's where you guys laugh, but you didn't. That's okay. That's cool. All right, so before I move on, are there any questions on the histone code, histone modifications, or DNA methylation? Yes.
You said that what histone, it's, what is it, acetyl something or other? It changes the charge of the histone.
It neutralizes the charge. So, and the example was a lysine being acetylated. And so that acetyl group being added neutralizes the charge there. Phosphorylation, phosphate groups are usually pretty negatively charged, so that'll add a negative charge. And so that's, those are meant to try and help explain how the nature by which these modifications can change the chromatin accessibility. It's changing the charge, thus it's like either promoting repulsion or promoting compaction, like a better phrase.
Phosphorylation has a negative charge.
It adds a negative charge, which some cases opens it up, in some cases it doesn't. Yeah. I'm gonna go to Nida, Justin, and then Tommy.
So the like acetylation, phosphorylation, all that's happening on amino acids, right?
Yes, on the amino acids on the histone.
So the histone itself is getting modified?
Yes, the histone is a protein that's being modified with the addition of these either acetyl groups, methyl groups, or phosphates. And that is, those small modifications can have repercussions that actually change the chromatin accessibility that, as a consequence, will change genes being accessible and the genes being transcribed.
So is there like a default activation level for a lot of these things? Like, do we need modifications to activate them, or yeah, what's the status quo?
It varies based on each gene. Each gene is going to be regulated very differently. And how some genes are regulated, we're still trying to understand. But there are some genes that always need to be expressed. We always need actin to be expressed in our cells. We need glucose transporters expressed all the time. But then there are other genes that don't need to be expressed all the time. You don't want your cells to be constantly expressing inflammatory proteins that can yield a strong immune response. And so how all of these genes are regulated differently at different time scales and in different contexts and circumstances. Cool. Genetics is hard. If this feels like this is a lot, it's because it is. That's okay. Yes.
So, patients are on like specific acids, like acetylation have context.
I'm gonna say no. No, that's a good question, because I feel like I only hear or read about, for acetylation, I feel like I only read about lysine. Methylation happens on a whole host of them. I'm gonna double check about that. So like, this is a great example of like, I should know this answer. I don't. I'll get back to you on that. All right, in the interest of time, let's keep going. Bless you. All right, so the last form of extra dogma regulation that will help set the stage for our the two syndromes that we're going to be talking about at the end today are going to be non-coding RNAs that regulate both at the DNA to RNA level, as well as most importantly, the RNA to protein level. So non-coding RNAs are regions of our genome that have been transcribed to make an RNA, but it's not translated into a protein. So this is a large class of RNAs that we call non-coding RNAs. Up to this point, when we talked about RNA in the central dogma, we primarily focus on mRNA because that's the class of RNA that is translated to make protein. But if you look at total RNA, either by mass or by the number of molecules in a cell, mRNA makes up a fairly small proportion of our cellular RNA. And so the two classes that we're going to be talking about in particular today, among our non-coding RNA, are going to be microRNAs and long non-coding RNAs. So we've already talked a little bit about a couple classes of non-coding RNAs, but are still very important and provide a really critical functional biological role, are ribosomal RNAs and transfer RNAs. We're all intimately familiar with those two now since we wrapped up Unit 3. A couple more are going to be microRNAs. MicroRNAs are these really small non-coding RNAs that actually will have a very sequence-specific downregulation of mRNA transcripts, meaning that they're going to be complementary to some specific mRNAs, then that they will then interfere with and halt their translation and promote their degradation. That process of blocking mRNA transcripts from being translated and promoting their degradation is called RNA interference. That was an entire finding that won the Nobel Prize a couple decades back. So the next class of non-coding RNAs that we're going to be focusing on today are, long non-coding RNAs. They are, while microRNAs are much smaller, on the order of 20 nucleotides long, 21 nucleotides long, long non-coding RNAs are much more massive and can actually act as what we call an RNA sponge, meaning that they, via base pairing, they're going to be, lack of a phrase, like soaking up RNA and inhibiting their activity, but also because of their large secondary structure, and we'll see what that looks like shortly, they're actually able to modify and act at the protein level and have a protein-like role in the cell and alter DNA accessibility. A couple more, so we have these small non-coding RNAs that encode for components of the spliceosome, the SNRPs, and we have SNOR RNA that facilitate modification of other protein molecules. But these are different classes of non-coding RNAs. While they don't code for a specific protein, they still have really important biological roles and are very functional. So first, we're going to talk about microRNA and RNA interference, what that looks like. So we have our pre-microRNA that needs to be processed here. And so there's going to be 3 key enzymes that are involved with RNA interference. So I'm going to ask you all to note here. The first one is DROSHA. DROSHA comes in and it will actually clip off the ends, including the cap here. And so from that, we get this microRNA hairpin loop. So it's going to be, after that, it's going to be exported from the nucleus out to the cytoplasm. The next enzyme that's going to be working here is called dicer. Dicer will cut off the loop at the end here and mature the microRNA into a mature duplex here. And so what ends up happening now is that historically, we only thought that one end of this duplex, one strand of the duplex, was going to be critical for RNA interference. So we used to call them a passenger strand and a guide strand. We've actually learned that both strands are able to both target mRNA transcripts. And so now we actually refer to them as the 5P arm and the 3P arm. And so whichever arm is going to be used to target an mRNA transcript, it's going to be loaded into this silencing complex with this protein called argonaute. And so once we have our specific strand loaded into argonaute, that's going to be called RISC, the RNA-induced silencing complex. So now what's going to end up happening is that if it has really good complementarity with an mRNA transcript, what's going to end up happening is that it's going to cleave that mRNA transcript. So now the poly-A tail, all of the pieces of a mature mRNA that helps it to avoid degradation by an exoribonuclease, now it is totally vulnerable to degradation. And so when there's really strong pairing here, the RISC complex is going to cleave it and promote degradation of that mRNA transcript. Another alternative option is if there's only if there's a less complementarity between the microRNA and the mRNA transcript, if there's less base pairing, what will end up happening is it's going to promote translational repression. It's going to stop ribosomes from going down the mRNA transcript and inhibiting translation of those regions that it's targeting here. And so we have our three key enzymes that I want you to take note of here: drosha, dicer, and argonaute. Argonaute with the microRNA forms RISC, the RNA-induced silencing complex. And we can either have strong base pairing that promotes cleaving and degradation, or less tight pairing that will repress translation. What's really great is that in a research setting, we've been able to use this RNA interference and leverage it to specifically target mRNA transcripts that we want to try and knock down and silence. Most of, I'm going to age a lot of the professors in the department. Most of the professors, when they were back when they were in grad school, like, this was not a resource available to them, but it's something we have now. And so it's a really powerful tool. And there's a couple different ways that we can go about doing this. One is an siRNA, whereby we start with a double-stranded RNA duplex that can specifically target an mRNA transcript of interest. It'll then be processed where we have a single strand that then will be loaded into Argonaute to then target an mRNA transcript of interest. And because we're either designing that siRNA, and I should say siRNA is small interfering RNA. Because you're designing it or picking it out yourself, there's going to be really good pairing, and that'll promote the cleaving and degradation of that mRNA transcript to suppress that protein being expressed. Another one is a short hairpin RNA, or shRNA. What ends up happening here is that you can have a DNA plasmid, or like a circular DNA construct that is under control of RNA polymerase III, the tRNA promoter, and will promote the transcription of the short hairpin RNA. It's going to be, the loop will be clipped, and we basically are up here where we were with siRNA. And so these are tools that we've developed that leverage RNA interference to try and target specific mRNAs to totally suppress their expression. Are there any questions here before we move on to our final concept before we wrap things up? Yes. I expect you to know these as example. I expect you to know that these are examples, not examples. These are tools that have been developed in light of our understanding of RNA interference. Yes.
I asked how these are introduced to.
Oh, yes. Sorry. Yes. I should have said that. Go ahead.
This type of organism or in vivo, how is this introduced?
Yeah, that's a great question. So these are introduced in a couple different ways. So you can either transfect cells with an siRNA. The key thing to note here is that RNA is pretty unstable. You know, there are exoribonucleases that want to degrade it. There's enzymes that degrade RNA, but typically you disrupt the cell membrane somehow to enable siRNA to go in. That's called transfection. So you can either transfect the siRNA with the siRNA, or you can transfect with the DNA plasmid, that DNA construct that enables you to leverage RNA polymerase III, so it's constantly cranking out the shRNA. And so the siRNA is it's a transient way of suppressing mRNA transcripts, whereas if you transfect with the DNA plasmid, that's a more stable transfection that'll enable you to knock down expression of whatever you're targeting. Great question. I should have said that. I'm glad you asked that. Yes. SH is short hairpin RNA, and DS is double-stranded RNA. A key thing to note here is that it's because it's double-stranded RNA, it needs to be fairly short, like 21 nucleotides, because double-stranded RNA that's any longer will mimic a viral infection, and you'll get a strong you'll elicit a strong immune response or a strong response because of that. And so we try and typically keep these on the shorter end. Both great questions, both things I should have tackled. Thanks for asking those questions. All right, so we're going to wrap it up with the last concept here. So with long non-coding RNAs, this is an arbitrary cutoff, but they are hundreds of nucleotides long. Whereas again, we were just talking about how our shRNA, siRNA, or in microRNA on the order of 20 nucleotides long. It's, excuse me, similar to mRNA. It's transcribed by RNA pol 2, 5' cap spliced and polyadenylated. But again, the key thing to note here, it does not encode for a protein. As it says in the name, it is a long non-coding RNA. So whenever, I should have said this already, whenever you hear it's non-coding, that means it does not encode for a protein. Long non-coding RNAs have a variety of functions. Among them is the ability to regulate protein activity by nature of long non-coding RNAs large secondary structure, which I think I should have. There we go. By nature of its large secondary structure, it can actually alter and regulate protein activity. Another thing is that it can enable post-transcriptional control. So like after transcription is occurring, it can compete and regulate microRNAs or mRNAs themselves. And finally, it can enable direct transcriptional control, which is exactly what we're going to use to understand Klinefelter syndrome and Down syndrome a little bit more. The way in which this happens is that there's going to be a specific long non-coding RNA that we're going to talk about that's called exist. So typically we have either people have an XY chromosome or an XX chromosome. And so there's a gene dosage problem because typically having two X chromosomes means you're going to have double the amount of protein that's encoded on the X chromosome. And so every copy number just typically means number of transcripts that are elicited from the number of copies you have of that chromosome. And when we talk about X-linked genes, that means genes on the X chromosome. What ends up happening is that there needs to be a compensatory mechanism so that there's constantly not always twice the copy number simply because a person has two X chromosomes. So the way in which the evolutionary strategy that's emerged to compensate for that is called X inactivation. What that means is typically during early development, one of the X chromosomes is randomly and permanently inactivated. We refer to that inactivated X chromosome as XI. That means the X-linked gene expression that occurs from proteins that are encoded on the X chromosome, those are encoded by the active X chromosome, which we call XA. And so to really nail home this point, one of these X chromosomes, when there's two of them, is going to be permanently and also importantly randomly inactivated. So that means in every cell, there's going to be a random between the two 50-50 chance, a random decision of which X chromosome is going to be inactivated here. And so this inactivation is mediated by a long non-coding RNA that's called exist, the X inactive specific transcript. And so the way in which this happens here is that exist, it's encoded for on the X chromosome. So when that long non-coding RNA is transcribed, exist will immediately go over and coat the X chromosome by binding to these exist binding sites along the X chromosome. What ends up happening is that as it binds, exist will actually begin to recruit at that locus a bunch of other proteins that'll promote DNA methylation to silence it. And then there's also going to be histomethylation. And then there's going to be this general just barring off of that inactivated X chromosome that it forms this heterochromatin that we call a bar body. And so if you stain for exist, you can actually see that off to the side within the nucleus, that X chromosome has been inactivated and has been segregated off into what's called a bar body. And so X-cis literally, it's encoded for on the X chromosome. Once it's transcribed, goes over and binds on these X-cis binding sites, recruits proteins that mediate the methylation at both the DNA and histone level, that then eventually will promote its curling up and balling, segregating off. Now, how is X-linked activation random? It's going to be, what ends up happening is that on the, there's an antisense long non-coding RNA called T6, just X-cis backwards, and antagonizes the actions of X-cis. So what that means is that the inactivated chromosome is going to be defined by X-cis coding all over it and the silencing of T6. So X-cis and T6 are going to be antagonizing one another. The inactivated chromosome is going to silence T6 and express X-cis, and it's going to be vice versa on the active chromosome. The active chromosome is going to be expressing T6 and silencing X-cis. And what that looks like is that there's going to be, each chromosome is going to be encoding for one and suppressing the other. And again, the chromosome that's being inactivated is going to be defined by, it's random, but it's going to be expressing X-cis. There's going to be a post-lecture video that I'll post that provides a bit of a mathematical explanation that helps us appreciate a little bit more about the randomness of this. But the key thing to note is that it's random, but the way we observe it is by looking at X-cis expression. So taking all of this back to Klinefelter and Down syndrome. And Klinefelter syndrome, again, just to remember that there's an extra X chromosome there. And what's key here is that that extra X chromosome is going to undergo X-linked X inactivation. And so that's going to silence the extra X chromosome. With Down syndrome, there's that third copy of chromosome 21. There's no mechanism in place to inactivate that extra chromosome. And so this is part of the reason that explains why there's a greater degree of severity with Down syndrome as opposed to Klinefelter syndrome. This X-linked inactivation is a way to deal with and handle that extra copy of the chromosome. But for other chromosomal disorders where there are extra copies, other aneuploidy disorders, there is no mechanism to deal with them. And so another example is Edwards syndrome, also called trisomy 18. It's 3 copies of chromosome 18. And it is characterized, symptomatically characterizes newborns with microcephaly, smaller heads, defects in many organs, and usually less than 10% of them live past to be a year old. And so typically, aneuploidy disorders are that are not X-linked can be quite deadly. So final thing, last couple slides before we finish up. The actual implications of X-linked inactivation. What that means is, again, I described before that there's a random decision between each cell which X chromosome is going to be inactivated. So that means in all females that they're all a bit of a mosaic, which meaning because different cells have different X chromosomes that have been silenced, which means there are different copies of X chromosomes that are expressed throughout the body. And so in one example of this in mammals is the fur color on cats is an X-linked gene. And so different X chromosomes have been inactivated across the body of the cat, which explains why they have sometimes different colors throughout their body. And so that's one example of mosaic expression of X chromosomes. Hemophilia is an X-linked mutation in clotting factors. It's overwhelmingly, males are overwhelmingly the hemophilic patients because they have one copy of that X chromosome. So if there's a mutation on it, they're more likely, they're going to express that mutated copy. Whereas in females, if they have 2 copies, there's a random inactivation of that mutated copy so that there's a 50% chance that they'll be expressing the mutated clotting factors. Finally, Rett syndrome, it's a somatic X-linked mutation, which means it doesn't happen on the germline. that affects paternal MECP2. That's a binding protein for methylated CpG DNA. And typically, because it's X-linked, only females have the copies whereby males will die in utero or shortly after birth. And so it typically doesn't present, because it's somatic, the Rett syndrome doesn't typically present in its severity until later in life. And depicted here is the same patient. And finally, proof of concept of a chromosome therapy to help tackle Down syndrome would be whereby we try to use X-cyst and X-linked activation. So if we took a fibroblast, we talked about that during the ECM lecture, it's just your ECM producing cell. If we're able to take any cell and basically make it into an embryonic stem cell via induced pluripotency, where we express certain transcription factors. If it was a trisomy 21 fibroblast, we could go ahead and use some of the same mechanisms that we talked about with Cas9, where we will try and target extra copy of a chromosome 21 and knock in an X-cyst binding site that'll enable us to deliver X-cyst that'll then inactivate one of the extra copies of the chromosome 21 that'll then ward it off into a bar body. And a group did that and published it in the best journal in the world and showed that that was a way to try and inactivate one of the extra copies of chromosome 21 in Down syndrome. If epigenetics really fascinates you, we have a great group of people here at UVA, one of whom, Nathan Sheffield's in our department. Given that I'm over time, I'll stop the recording here and hang out up here for questions. Thank you.